like to welcome everyone to this Adelaide Biomed City Research Mini Review session with a specific focus on RNA therapies. I'd like first to acknowledge that most of us today are meeting on land that is where the Ghana people are the traditional custodians, and to pay our respects to their elders past, present and emerging. I'd also like to remind you that the session is being recorded and will be available later on the ABMC website. Also like to remind you that if you've got any questions at the end of either of the two presentations, if you could insert those into the Q&A section in the Zoom session. I suppose I should introduce myself as well. Sorry, my name's Boddy. I'm the Dean of Research in Clinical and Health Sciences at UniSA. So our first speaker today is Associate Professor Cameron Bracken, who works in the Centre for Cancer Biology here at UniSA, and his talk is going to be on targeting cancer with RNA therapy. Over to you, Cameron.
Thanks for the intro, Alan. So yeah, I'll be talking to you today about new opportunities for RNA therapy and giving you a very short overview of the sort of things that historically we've done in the lab, and how it's led to hopefully a slightly more applied future outcome. So for those of you who are not aware, we're clearly not going to spend much time with what microRNAs are. Suffice to say that microRNAs are suppressors of gene expression, and the way that in which they work is that this is representing a protein called Argonaute, which binds and displays the microRNA. There is a small cleft where there are a short section of the microRNA which is exposed, and that's the region of the microRNA which does base pairing to its target mRNAs, and hence is responsible for targeting. This region is known as the seed sequence, which is toward the 5' ends. And the thing you will notice about it is it's really short. We're only talking sort of 6, 7, 8 nucleotides. And so you can imagine that will occur frequently throughout the transcriptome. And so a lot of our work historically has been what makes something a target and something else not a target. Now, the ways microRNAs work, obviously, is that there are potentially thousands of target transcripts per microRNA because of the short nature of the binding interface. And so, of course, any one of these targets might have a function, particularly if something is a particularly strong target and that happens to play an important role like a transcription factor. By definition, that's a master regulator of processes downstream. And so these can, I guess, exacerbate sort of network effects. Or potentially microRNAs can also have an effect by being the sum of the parts. So although you may have a series of more modest effect on targets, if the targets participate in a common pathway or complex, then that can lead to quite strong biological outcomes nevertheless. So historically, our lab, Phil, Gregory, Greg Goodall as well, we've worked on a group of microRNAs called the MIR200 family. Don't be fooled by the name. Despite these, they're all part of the MIR200 family. They're encoded on 2 different chromosomes, and they're designated, in this case, green and brown, because they have different target affinities. And so cumulatively, these 5 microRNAs play a very, very important role in a process that we are interested in, which is called epithelial mesenchymal transition. And a lot of work that we've done in the past that started off with the second speaker today, Phil, is that the MIR200 family participates in a reciprocal feedback loop with a key transcription factor called ZEB. And so it's evidence of that particular type of targeting that we spoke about in the previous slide. Here you have these epithelial cells, which are immobile. If you want cells to move in development or in wound healing, then you can activate this process and turn them into a mesenchymal cell, which is mobile. And then that process can be reversible. Great for development, great for wound healing, not necessarily so excellent when you are looking at something like metastasis or chemoresistance. Now, we have also done a fair bit of work on this other mode of microRNA binding where you have multiple targets within a common pathway. And in our particular case, we know that the MIR200 family hits a lot of cytoskeletal proteins and hits a lot of genes associated with the EGF signaling pathway, and that has various metastatic outcomes as well. So in an effort to try and move this, I guess, toward a a more practical real-world outcome. Small RNAs are an opportunity for therapy, particularly because, at least in theory, one can target any gene that one desires with a small RNA. So the problem is that for any one microRNA to have a function or to have a strong effect, you often need a very high concentration. But the problem, of course, is when you've got a high concentration, there is a lot of off-target effects that will occur and ultimately toxicity. So the solution perhaps for this, perhaps a better type of RNA therapy, comes with a study that we published a couple of years ago, where we found not just the miR200 family, but there's actually a range of these microRNAs that are all different, but they participate together and they synergize to drive this reverse MET process. We're not going to go through the data today, but suffice to say that at very, very low concentrations, or essentially endogenous concentrations, any one of these microRNAs does very, very little toward, in this case, MET. However, when you combine them, they synergize at subnanomolar concentrations and they have quite a profound effect. And so we hypothesized perhaps that by combining these small RNAs, we may have a therapeutic window of opportunity where we can maximize an effect and minimize concentration and ultimately toxicity. We've applied this initially to the Rast signaling pathway because this is fundamentally important in various cancers. Rast signaling is a cascade of 10 kinases which are regulated by different genes. Ultimately, it promotes proliferation and survival of cells. Importantly, when we have all of the, in this case, siRNAs, not microRNAs, but essentially something very similar, we can knock down these individual components of the signaling pathway, but you really need to knock down all of them to have quite a profound effect on the output of the signaling pathway. And when you actually look at what this does to cell behavior, it gets very interesting because in cells where the Rast pathway is not oncogenic, it's just operating at natural levels, then the combination of all of these siRNAs does nothing to cell viability. However, in particularly melanoma cells, which are surviving or proliferating because of overexpressed Rast signaling due to a mutation in the key kinase, then these siRNAs decrease cell viability quite profoundly. Importantly, you need all of the siRNAs to be working together. It's not sufficient at low concentrations to have any one siRNA. You actually need the combination to decrease viability. And perhaps most interestingly of all, in the context of something like melanoma, the problem with metastatic melanoma is that patients develop resistance to drugs like vemurafenib. And so with a melanoma cell line which is sensitive to this drug, then they are obviously drug sensitive and they're affected by the siRNAs. Importantly, however, in the cells which have become resistant, which they do in patients almost invariably, they are no longer sensitive to the drug, but they're still sensitive to the siRNA. And so the hope is that perhaps by combining these siRNAs, we can actually target a stage of cancer which is otherwise resistant to current therapies. The very last thing that I'll leave on is that with siRNAs, they're terrific, except they are slightly inhibited by inflexibility in terms of what you're able to design siRNAs against and some of the background effects that might occur. And so we've also been doing some work with, can we actually synthesize entirely artificial microRNAs? And what we find is that we can develop a number of completely artificial microRNAs that will target 4, 5, 6 members of the Rast pathway or any of these other oncogenic pathways. And they do so a lot more highly than they do background targeting. And so we think that these artificial microRNAs might be an even better way still to try and exploit that therapeutic window between effect at low concentrations and avoiding sort of toxicity. So all that remains in the very short talk is to thank the people who have actually done the work, and that's primarily Melody in the lab and Kat in bioinformatics. And thank you for your attendance. Okay. Alan, I can't hear you.
Yeah, thank you, Cameron. Thank you for pointing that out. First lesson of doing anything on Zoom, don't mute yourself. Thanks, that was a great presentation and certainly rattled through some of the complexity of bringing these potential new therapeutics to bear on, as you say, particularly challenging types of cancer. There aren't any questions fielded through the Q&A or the chat, but can I ask a technical question? The synthetic microRNAs, are they conventional nucleotides still? or are you putting in artificial nucleotides as well?
Yeah, that's a good point. So the nucleotides, the sequence themselves are conventional, but there are a lot of modifications that you get to in terms of RNA therapy. There's a lot of modifications that you can put onto the nucleotides to increase things like stability and ARGO interaction. And so ultimately, when we use in vivo models, they will be modified, but the nucleotides themselves are the sort of nature that you'd expect them to be.
Right, and then are there additional challenges as well? Because no one else is asking a question, so I'll keep going. In terms of getting these new molecules, these new potential therapeutics into cells and whether they need to act within the cell as well.
Yeah. So the real challenge with the good thing with RNA therapy is that in the space of 10 minutes, you can design an RNA that will target anything, which is great. One of the big problems, of course, is delivering it to where it needs to go. At the moment, that's a significant challenge with the exception of the liver. In the liver, there's a sugar molecule that you can directly attach, and that's being used as a basis for the first 3 siRNA therapies that have been approved for, well, specifically treat liver diseases. The sugar is uptaken by hepatocytes really effectively, and it goes there extremely well. The challenge remains with things that's not hepatocytes. And so people have tricks about targeting things like EGF receptors and stuff on the surface of cells, but there's a lot of development that needs to be done in that space to really hone in on that cell specificity. Okay,
but as you say, the attraction is the potential of these things as potential new ways to target cancer. So that's really good to see. Okay, thanks, Cameron. We'll move on to our second speaker, Associate Professor Phil Gregory, also from CCB. The title of Phil's talk is MicroRNAs, Small but Powerful Regulators of Breast Cancer Progression. Over to you, Phil, if you can share.
Well, thanks for this invitation to speak today and for your words, Alan. So in the short time I have, I'm going to talk a little bit about a project that we have in breast cancer, specifically metastatic progression, and I'll give a bit of an introduction to breast cancer, but I'll keep that short in the interest of time. So as we all know, breast cancer is a very prevalent disease, and despite a really great amount of new treatments coming forward, unfortunately about 8 women in Australia die every day still from breast cancer. And we know that breast cancer is a very heterogeneous disease, and it's really when it becomes that aggressive stage 4 disease where you see a lot of metastases that the survival of women really plummets, and it's only about 30% women that have this metastatic disease that survive greater than 5 years. So clearly there is a great need to understand the process of breast cancer metastasis. And our work is really focused on a subtype of breast cancer called triple-negative breast cancers. Now these account for about 15 to 20% of all breast cancers, and they unfortunately are one of the most aggressive types of breast cancers. So they have very poor prognosis, and there is very limited therapies available to treat these types of breast cancers. So women that come in with triple-negative breast cancers essentially receive chemotherapy, and while this helps in the short term, the cancers invariably relapse, and unfortunately many women succumb to metastatic disease. And so one of our projects involved a collaboration with Professor Robin Anderson at the Olivia Newton-John, and this was started many years ago, and like many good projects, it started with a screen. And so this screen involved using a series of mouse breast cancer cell lines. They're isogenic cell lines, so they're all derived from the same primary tumour, but they have different propensities to metastasize. So you can see that some can form primary tumours like the 67NR but can't metastasize, through to other cell lines which have very striking metastatic abilities. And so we were interested in the possibility that microRNAs might regulate this process. And so we profiled microRNA expression within tumours derived from this model. And you can see here with the graph on the left, a number of microRNAs changed, and we were specifically interested in one called MIR342, primarily because nothing was known about MIR342, but also, as you can see from the graphs on the right, that MIR342 is very strongly repressed in the more metastatic cells. So this suggested that perhaps this microRNA might act as a suppressor of triple-negative breast cancer metastasis. When we looked in patient data, this also supported this potential role as a tumour suppressor. So we saw that in triple-negative breast cancers, that tumours with particularly low MIR342 levels had very poor survival. This was also similar in tumours that were ER negative, so not quite triple-negative, but they've already lost estrogen receptor expression. However, in the estrogen receptor positive breast cancers, we didn't really see this trend. So this suggested that perhaps specifically in triple negative and ER negative breast cancers, that MIR342 might act as a tumour suppressor or a metastasis suppressor. So we went on to carry out some in vivo studies. So this is an example of some of the types of analyses that we do in our lab. So we took the very metastatic mouse breast cancer cells and we essentially re-expressed MIR342 back into these cells. And then we implanted these into the mammary fat pad of mice, where afterwards they can spontaneously metastasize to different organs. And as you can see, we found that MIR342 significantly shrunk these tumours, so they grew at a slower rate, but also the metastases to various organs was markedly reduced, as you can see in the graphs depicted there. So we next wanted to see whether this could extend to human breast cancer cells. And we used a human breast cancer cell line that had very low levels of MIR342 and was highly metastatic. And in the same way, we put MIR342 back into these cells and then grew these cells in the mammary fat pad of mice as tumours. In this case, what we observed was something a little bit different. The tumours themselves grew at essentially the same rate, but we saw a marked reduction in the amount of metastases to different organs. So you can see that is represented by smaller lymph nodes, but also much less metastases to the lung and to the liver. And so collectively, this really suggested that both in human and mouse cells, in both an immune competent and an immune deficient environment, that MIR342 was acting as a suppressor of metastasis. So I guess the next obvious question is how is it doing that? And so we do this quite commonly in the lab, an RNA-seq analysis, followed by gene set enrichment analysis to try and work out which pathways MIR342 might be regulating. And what we found was that MIR342 strongly suppressed the E2F signaling pathway. Now, for those who know a little bit about breast cancer, E2F signaling is very important in driving the growth of cancer cells, and in particular is known as a driver of ER-positive breast cancer. And so there's already drugs out there which try to target E2F signaling to limit the growth of, in particular, ER-positive breast cancers. So knowing this, we also looked to see where MIR342 was specifically binding. And so to do that, we used a technique called ClipSeq, where we can pull out all the 342 binding sites in the transcriptome and then subject them to deep sequencing to find out exactly where they're binding. And we found that MIR342 bound to roughly 120 specific mRNAs. And when we looked at what those mRNAs were, we found that E2F1 itself, so the transcription factor that drives E2F signaling, as well as many E2F1 target genes, were very strongly targeted by MIR342, suggesting that it acts through direct and indirect mechanisms to strongly repress the E2F signaling pathway. So the next question was, well, is this relevant for human breast cancers? So to check that, we took a very large set of triple negative breast cancers, and these were divided into tumours that had high or low MIR342 expression. And we asked, well, what signatures can we see that are different between these two groups? And again, what we saw was that the E2F signaling pathway was strongly repressed, specifically in the cancers that had very high expression of MIR342. So this suggests that what we observed in our screen and in our mouse models was also represented in human clinical samples. And so I'll just finish here with our working model. We're hoping to publish this work soon. So MIR342 we found suppresses the E2F1 transcription factor, but also at the same time represses a number of downstream targets at E2F. And we think that this dual nature of targeting results in a strong dampening of this pathway. And our hypothesis is that in subsets of triple negative breast cancers with low MIR342 expression and therefore high E2F signaling, that the high E2F signaling is actually leading to a more aggressive cancer and its ability to spread to multiple organs. So one of the interesting aspects of this is, as I mentioned earlier, there are drugs available such as palbociclib, which can target the upstream kinases which activate E2F signaling, and potentially these types of drugs may be useful to limit the metastatic ability of triple negative breast cancers. And this is an indication that is only really beginning to be explored in terms of their therapeutic utility. So this is an area that we're also exploring using mouse models and hopefully in human tumours shortly as well. So that just leaves me to thank members of my lab and the Centre for Cancer Biology who have all contributed to this project in many ways. We're very well supported by the ACRF Cancer Genomics Facility based here at CCB. In particular, Dr. Catherine Pillman contributed substantially to this work, as did John Tubia. We have a lot of collaborators that have been involved in this work over the years, and just like to thank the funding bodies and thank you for your attention.
Okay, brilliant. Thanks very much, Phil. And certainly a very interesting presentation that kind of complements Cameron's previous presentation as well. Just looking in the Q&A, I don't think there's any questions directly in there. I mean, again, I can come up with a question. So around those CDK inhibitors, do you see this as something that is a complementary treatment? The RNA-based approach would be in combination with those CDK inhibitors, or is this something where they're given sequentially? What is it, a question of resistance that might be overcome by the RNA?
Yeah, that's yes, that both potentially are options. You could imagine that MIR342 could be by itself a single agent that would help overcome some resistance that arises. But I think what we've discovered by looking at the actions of MIR342 is potentially the ability to use a drug which is already in clinical use and is safe, and potentially if we're able to subgroup types of triple negative breast cancers, we may be able to treat those particular subtypes with this drug and potentially, limit their ability to metastasize and outgrow, which would be a huge improvement for this disease. Okay, that's brilliant. So it's kind of a dual role for the RNAs. I've got a question I'll put to both of you if you don't mind, and we'll see if this real ignorant question can generate any interesting answer. Why are these microRNAs now coming to the fore? I mean, it used to be RNA, DNA, and kind of knew where I was. Is this a whole new area of understanding the biology of cells and how they work? And there's many more of these, I understand, microRNAs. We're still teasing those out. Yeah, well, I'll start. So I think, microRNAs, so they've been known for sort of 20 odd years, but it's only really been the last decade that we've sort of began to understand what they do. And I think there's certain microRNAs that they're sort of powerful drivers of cell fates, like Cameron shared earlier. And then there's other ones which sort of act a bit more like buffers of systems. So they in themselves, they sort of maintain the homeostasis of cells, but perhaps they're not involved in these sort of radical cell transitions and potentially may not be as helpful in a therapeutic sense. But I think it is an exciting time. I mean, everyone's talking about RNA therapies with all the COVID vaccines and sort of all the technology to be able to deliver these RNAs is there and available. So it's just a matter of sort of optimizing delivery to various locations and then finding out, which diseases be most amenable to treatment.
Yeah, that's great to hear. And as you say, you've got that almost sort of proof of concept, massive experiment with billions of people being given an RNA as a therapeutic now. And,
yeah, that's right. That's right.
Okay, brilliant. Thank you very much, both of you. As I say, there don't seem to be any questions from the audience, but you can continue to provide feedback, comments on the Q&A function for the next few minutes if you want, or you can contact the Adelaide Biomed City website if you've got any comments on today's presentations. The presentation will be uploaded to that website at some point in the future. Just leaves me to thank today's presenters, Phil and Cameron. Next week's presentation will be on obesity with presentations by Leonie Heilbronn and Tongji Wu, chaired by Andrew Zanatino. So thank you all for attending, and I hope to see you, and I'll just be a participant then on this channel next week. Okay, bye.